welcome to the online lecture. We will continue to talk about chromosome and the human genetic disorder. We will first start with some review of what we learned from last week, and then we'll move on to human genetic disorder. Specifically, today we will focus on the sex-linked disorder. So let's review some basic concept. Each chromosome, they have many alleles, and the allele they are located in the location of the chromosome. We call them locus. And depending on the location, depending on the locus of the allele, if the locus of two alleles they are close to each other, we call it linked gene. But if there are multiple alleles that they are located very close to each other, we call them linkage group. And then if the alleles they are linked, then it will be difficult for crossing over to occur. Remember, crossing over occur in which stage? In meiosis? Yes, let's think about this. Meiosis, we have meiosis 1, meiosis 2. And it occurs in meiosis 1, and then which stage? In meiosis 1. If you remember my lecture, crossing over, or we call it recombination, occur in prophase 1. So in prophase 1, when crossing over occur, usually it occurs when the exchange of genetic material that are in only certain part of the chromosome. So if the gene, if the allele they are linked together, that means the locus or the locus is very close to each other. It will be very difficult to separate the two alleles during the crossover. In other words, if crossover occur, the two alleles they will either stay in the same chromosome, in the same chromatid, or they will move together to the different chromatid. Because they are so close together, they're linked. So this is an example of all kinds of allele, or the trait or the disorder that you may have eye color, hair color, and then you have all the deficit or disorder, deafness or anemia. They all in the same chromosome, and also they are in the, for example, this part they are close to each other. When crossing over occurs, then all these alleles they may move together, or they will stay at the same chromatid. This is an example. So when crossing over occur in prophase 1 during meiosis, so A and B, because the allele A and allele B, because they are close to each other, they will stay to each other instead of moving. So only the top part of the allele they will do the crossover exchange between the chromatid. As a review, you have to know that chromatid, the crossing over is the exchange of genetic material between chromatids. So, and also at the end of mitosis, at the end of meiosis, you will produce 4 daughter cells, and they are gametes, instead of 2 daughter cells. Producing 2 daughter cells is mitosis. Producing 4 daughter cells is meiosis. And most importantly, crossing over occur between the distance gene. If the 2 genes they are located very close to each other, when they are linked, it will be difficult to have crossing over occur. This is another example. So gene A and gene B, they are located farther away from each other, one on the top, one at the bottom. So what happens is that when crossing over occur, then the gene B, they can exchange because it is not linked to gene A. We call it recombination because there's an exchange of genetic material. So genes that are farther apart, then the chances of having crossing over will be higher. It will be easier to have crossing over. So we have 23 pair of chromosomes, 22 pair recorded autosomes, and then 1 pair recorded sex chromosomes. The 23rd pair. The sex chromosome, again, we have for male it is XY, for female it is XX. And X and Y, they contain different combination of genes. That's why we do not consider them as homologous. Because Y chromosome is a lot smaller, and it is a lot, it carries only a few alleles. And sex chromosome, they also have sex-linked trait. Most of the sex-linked trait, they are located on the X chromosome. The reason is that Y chromosome is very short. They only have a few alleles. So basically, to simplify in our class, you can assume that Y chromosome is genetically empty. So you will assume that Y chromosome does not carry any allele gene, just to simplify our problem. Most of the gene allele, they are carried by the X chromosome. For male, we have X and Y chromosomes. The X chromosome in male is carried by, the X chromosome in male is inherited from the mother. The Y chromosome from a male is inherited from the father. And most of the sex-linked genetic disorder, they are recessive. For female, they must receive 2 recessive alleles in order to have that genetic disorder. For male, they only need 1 recessive allele from the X chromosome from their mother, and then they will already express their genetic disorder. One example, we talked about it last week. One example is that for the color blindness, it is a sex-linked disorder. If the X chromosome have this uppercase B, that this allele, that means it's normal vision. If this is a lowercase b, then it is a recessive allele, then we have the color blindness. Most of the color blindness is red and green color blindness, and it is a very common X-linked disorder. For carrier means that female is heterozygous dominant, meaning that the female has 1 dominant allele and 1 recessive allele. Then we call this carrier, heterozygous dominant. For colorblind female, it is homozygous recessive, meaning that the female carries 2 recessive alleles. And for the healthy or normal female, that means the female has 2 dominant alleles. We have more colorblind male than colorblind female. The reason is that for colorblind male, we only need 1 recessive allele, and then on the X chromosome, and then the male will have color blindness. For female, in order to have color blindness, the female needs 2 recessive alleles, 1 in each X chromosome. Then the female will develop color blindness. That's why, in terms of percentage, we have more male to have color blind than female. So these are the cases. For normal, healthy female, she will have 2 dominant alleles. For carrier female, she will have 1 dominant and 1 recessive allele. For female who is colorblind, she will need to have two recessive alleles. But for male, we only have two cases. It's either normal or colorblind. Normal male, it will have one dominant allele on the X chromosome. Because Y chromosome, we assume that it is empty. For male colorblind, you only need to have one recessive allele on the X chromosome. In other words, male does not have a carrier. Male does not have carrier because Y chromosome is empty. So I want you to think about this question before you view the second video. So if a male with normal vision marry a heterozygous female, that means marry a carrier female, what will be the percentage of chances they have colorblind daughter and a colorblind son? Okay, I want you to draw a Punnett square and try to answer this question, and then I'll tell you the answer after in the second video.